Assalamualaikum. Hello everyone. It is my great pleasure to talk to you today. My name is Dr. El-Wafa. I have been an assistant professor in biomedical engineering department, Minya University, Egypt, for over 10 years. I am so happy to be here today on behalf of my colleagues, Engineer Asma Slim, which is this paper is part of her master's degree in electronic engineering, and the Professor Hisham Hamid. Our presentation will be about hardware acceleration for the Smith-Waterman algorithm for short-read DNA alignment using FPGA. This is five parts in 30 slides. First part, introduction to bioinformatics science. Second part, explaining the problems facing the bioinformatics science. Third part and the fourth one, about our way to solve these problems and the results we got from our experiments. Then at the end, we will summarize our conclusion and future work. The bioinformatics science and gene banks in 7 slides. Is a science building bridges between the biology, computer science, pharmacology, biostatistics, and the more other sciences to solve the biological problems and the human diseases. The bioinformatics science nowadays is a major research field in pharmacology and biology. Is bioinformatics and DNA is so important? Bioinformatics science dealing with DNA, RNA, and amino acid, which influencing different sciences, as in medicine for cloning and detect diseases, in agriculture, in vaccines, and drug design as anti-cancer drug and chemotherapy. All of our genetic information from the color of our eyes to the color of our skin in every cell in our body. This genetic information is coded in 4 different letters named adenine A, cytosine C, guanine G, and thymine T. Example of FASTA format for liver genes and how it coded in combination of our 4 letters. Genome has been composed of 6 billion nucleotides of DNA. The nucleobasis is one of our 4 letters. We can get this information by having a sample of blood or a tissue, analyzing this sample in a machine called the DNA sequencer, which gives us a result of millions of small sequences. This process will take a few seconds, but to get a final sequence of this sample, it will take hours, days, or weeks using a special bioinformatics software. Reference sequence DNA of a known tissue, as an example, a kidney tissue, and we got unknown tissue DNA sequence by comparing both sequences by special software and have alignment between them. If the result got that they are the same, which means that they have the same function and the unknown tissue is from the kidney too. All the human genomes and most of the species genomes are stored in the 3 famous gene banks: NECBI at USA, ENA at Europe, NIAS in Japan. Billions and billions genetic data stored in the gene banks. In 2 slides, we define one of the biggest bioinformatics problems. The 4 problems we try to fix them are the huge data in the gene banks, which increase dramatically every day, causing a memory bottleneck for analyzing this data, and slowing the computational time for aligning this data too, which affecting the accuracy of the result. Our way to fix this problem is to find an accurate algorithm and find a way to accelerate the computational time. Curve, according to the NECBI Gene Bank's report release 210, showing that the data stored in the gene bank at October 2015 about 7 multiplied by 10 power 10 base pair, which means the data is tripled every 18 months. Informatics problems. We will answer this question in the third part in 12 slides using 2 ways. This slide will summarize all the bioinformatics sequence alignment algorithm, which is divided in 2 sections. The pairwise algorithm for aligning 2 sequences and the multiple sequence for more than 2. In the most right part, we'll find the 5 algorithm, which give us an approximate alignment result and the two slow algorithms, which give an optimal exact alignment, Smith-Waterman and Middleman Wunsch. Then we made a comparison between the most used algorithms in bioinformatics. We decided to choose a Smith-Waterman algorithm as the most accurate algorithm, with a disadvantage of long computational time. The algorithm consists of three steps. First step is the initialization, as if we have unknown sequences horizontally as a reference sequence and the query unknown reference sequence, the first step will initialize the first column and the first row with zeros. Then to start to complete every cell in the matrix until the last cell. Second step, we got the highest number in the at the high, which called the high score in this matrix. The last step, we tracing back from this highest number on to the first initialized cell, the optimal alignment. In this slide, we explain how to calculate every cell in the matrix. If you want to calculate this cell as an example, we need to know A and B and C, and we need to compare the two letters in the reference sequence and the query reference. If they match, we add A plus the match reward. If mismatch, we will add A plus mismatch penalty. Then we get the maximum number between A and match reward, or A plus mismatch penalty, or B minus the gap penalty, or C minus a gap penalty, or 0. Gap penalty is defined according to the insertion or deletion in sequence, which is called in biology a mutation. Summarize the three steps in Smith-Waterman algorithm. And here an example, we find the high score is 30, we're tracing back this cell to the previous cell to the previous cell until we find the last optimal alignment. Now we got Smith-Waterman as an algorithm to be used in our solution. We want to accelerate this. For the hardware implementation, we compared three options: personal computer, FPGAs, and ASICs. The FPGA as an intermediate solution and good performance for an affordable price. 7000 from Xilinx board and our hardware platform with a dual-core ARM Cortex A9 and 53 kilos LUTs. How to program our Zinc 7000 with a sequential Smith-Waterman algorithm VHDL code, where our design use 1 comparator, 6 adder, and 6 maxspice functions to get the value of each cell. We use the concord and extend techniques to parallelize our alignment as shown in the figure. We divide the reference sequence to subsequences. Every subsequence has a query length. Then we calculate all submatrix in parallel way. Then by the end, we combine the result to get the local and the global alignment in the same time. This slide our algorithm pseudocode. Step 1, we divide every reference sequences. Step 2, calculate the submatrices in the parallel process. Step 3, combine the result from each process to get the final alignment. This is the hardware design inside the Zinc 7000 FPGA, which consists of 5 blocks. Each block has individual VHDL codes and connected together by its input and outputs. First block is a baud rate 9600, which adjusts system clock 100 MHz to 9600 bits per second, which is the same rate of the transmitter UART. Second block is the transmitter UART block, responsible for transmitted data from Zinc 7000 to the PC. Third block is a parallel to serial 128 byte. This block converts data from parallel state to the serial state, so data compatible for the transmitter UART. Fourth part is a mapping block, which converts the result data output alignment from the digital number 0 1s to letters. to be readable and then send these letters to the barrel to serial block to become compatible for transmit UART. Last block is a Smith-Waterman alignment. It is our main block in our design, which does the Smith-Waterman algorithm calculation, as we explained before in the parallel process. It takes an input sequences and the final alignment result transmitted from FPGA to the PC. Our result in 2 slides. Our design simulation for the short read DNA references and the query sequences. The sequentials of Smith-Waterman algorithm and our parallel design in 2 subsequences and 3 subsequences, we find that the FPGA utilization is decreased from 18% to 1%, and the efficiency is increased by about 30 times, and GCAPS per watts is increased from 3.12 to 91.8, which explains that our new design using parallel hardware Smith-Waterman using divide and extend techniques more efficient and accurate with high speed. Next slide. Develop a new parallel hardware for Smith-Waterman algorithm based on divide and extend techniques, PHSW-DE. Our algorithm give a global and local alignment in the same time. Our algorithm doubles the speed of the alignment and decreasing the FPGA utilization. In the last, we'll talk about our future work. Try to upgrade our design for a multiple sequence alignment. Then we could try to use a cluster FPGA to speed up our alignment. We'll increase the sequence size to be aligned. And finally, we try to connect the cluster FPGA system to the sequencer directly. شكرا لكم. Thank you everybody for listening and care. Merci, danke, arigato, gozaimasu. This is my mail if you need any further information about our work.